So here in 18.4, we are going to take a look at B cells now. So we've talked all about T cells. We have also spoken about how T cells, the T helper cells, can help to activate the B cells. So now let's take a look at the B cells. Remember that the B cells are part of the humoral immunity, meaning that they float around in the humors, in the fluids of the body. Remember that they begin, divide, and mature in the bone marrow. That's why they are B cells, because they start out, divide, and mature in the bone marrow. Each B cell expresses a specific, randomly generated antibody on its surface. So just like we saw with the T cells, where the T cells go to the thymus, and then they are given this randomly generated receptor, a B cell also is given a randomly generated receptor. In this case, it's called an antibody, though, because remember we had those antibodies that are embedded in the plasma membrane. Just like you can see this image over here. Here's our B cell plasma membrane. Here's the antibody that's embedded in it, and then we have our antigen binding sites. These antigen binding sites, remember, are variable. They can change, and the way that they change, the way that each B cell has these antibodies embedded within them, is different. Randomly generated again. So the antibody acts as a receptor for a specific antigen. So B cell receptors are the BCRs. So similar to the T cells, except just a little bit different because it's actually an antibody that's embedded in the plasma membrane. Similar to the T cells, the B cells are then tested to make sure that that antibody does not bind to self-antigens, right? So again, it's tested still in the bone marrow to make sure that it doesn't bind to our self cells and say that they're bad, say that they're pathogens, say that they need to be destroyed, or whatever that particular antibody does. It doesn't say that it needs to be destroyed, but remember our antibodies, what they do are neutralization, opsonization. They're going to be activating our complement system and various other things we spoke about, agglutination, things like that. So our B cells are tested to make sure that they don't bind to our self cells, our self antigens. The B cells that pass are going to divide and then migrate to our lymphatic tissues and organs, just like we saw with the T cells. Of course, those that don't pass are destroyed. And then at this point, these cells are called immunocompetent and naive. So just like we saw with the T cells, except this is happening in the bone marrow, and the receptor is called an antibody. It's an embedded one in the plasma membrane. Versus the T cells, this happens in the thymus, and it's just a regular receptor. And then they all go out to the lymphatic tissues and the organs. So now the B cell receptors, so they are again immunocompetent and naive. The B cell receptors can interact with antigenic epitopes on free antigens or epitopes on the surface of an intact pathogen. So if we have our intact pathogen, right, so we have our bacteria that has gotten to the body somehow, our B cell receptors can interact with those intact pathogens. So it'll actually, if that B cell receptor, if the antibody on the B cell has the correct match on the end of the antibody to match up with the antigen on an intact pathogen, it will do that. Or if it is a free antigen or it's floating around, so say some bacteria have already been destroyed and then those free antigens are floating around in the fluid and it bumps into one of these B cells that are naive but it matches perfectly, then it will activate that cell. So on a free antigen, if it's floating around, or epitopes on the surface of an intact pathogen. Note that this is different than the T cells because remember the T cell receptors can only interact with antigenic epitopes that are presented on the MHC class 1 and MHC class 2 proteins. So a T cell, right, can't just be floating around or walking around through the tissues and then just come into contact with a pathogen and match up and then become activated. T cell receptors have to match up with that MHC class 1 or MHC class 2 molecule, and then they can go on to be activated and do what they do. B cell receptors, again, can interact with anything that's just floating around. So if it matches up with anything that's floating around, it does, or if it's on the actual pathogen itself and it matches up, then it does. Also, B cell receptors can interact with epitopes of all classes. So remember the proteins, the lipids, the carbohydrates, the nucleic acids. Those are the different classes of receptors, or of epitopes rather. Whereas our T cell receptors can only interact with protein epitopes. So we did speak about that briefly when we spoke about the different classes of epitopes. So our T cell receptors can only interact with protein epitopes. So even if an antigen presenting cell is presenting an antigen that is a carbohydrate, our T cell receptors are not going to recognize it. Our B cell receptors can recognize all of those different classes. So protein antigens then are T cell dependent antigens. So we can have a B cell, right, that's being used as an antigen presenting cell. Remember, we said that our MHC class 2 molecules were on macrophages, dendritic cells, and B cells. So if our B cell comes into contact with a free antigen or epitope, or it comes into contact with the surface pathogen or the surface, an epitope on the surface of the pathogen, then what it can do is it can act as an antigen presenting cell. It can act as an antigen presenting cell to the T helper cell. So it can present it along with the MHC class 2, and then it can present that to the T helper cell. Now, when we talk about polysaccharides, lipopolysaccharides, and other non-protein antigens, these are T cell independent antigens, so separate from the T cell. These do not depend on the T cell for activation. So we're going to take a look at that in an illustrated version here. So when we talk about T cell dependent activation of B cells, here's what we see. One, our B cell interacts with a pathogen or a free antigen, just like we said. So here, if we look at the illustration, here's the bacterium. It has the antigen on it with its little epitopes. Here is our B cell, which is naive at this point, but it happens to have the B cell receptor that matches up with the epitope on this particular antigen / bacterium. Once this happens, it is able to present that. You can see here, our B cell is the one on the left here. Here is the bacterium with the epitope. Then it's able to present that on the MHC class 2 molecule. So here is our little epitope saying this is what this guy looks like. Then we have our T cell receptor that's going to come along and match up with that. We have our CD4 receptor. This tells us it's a helper cell. We have our helper T cell come along, match up to that same piece, that little epitope. And what's going to happen is that this helper T cell is now going to release its cytokines. So remember, we said that that's one of the things the helper T cell does. We said the helper T cell activates our cytotoxic T cell, which we just saw a little bit ago. We also said that it's going to activate our nonspecific immune response, right? Because cytokines do that. They activate our inflammatory response in fever. We also said that when that T cell is activated, the helper T cell is activated and it releases those cytokines, it's going to activate the B cells. This is how it activates the B cells. The B cell first interacts with or encounters the pathogen. Then it's going to present a piece of that pathogen on its MHC class 2 molecule. At this point, let's forget about the bottom of the illustration right now. If it's just presenting it on the MHC class 2 and it has it attached over here, it is not an activated B cell at this point. It has to be activated by interacting with that T cell. So then we can see at the bottom here, it does that. It interacts with the T cell. That helper T cell then releases those cytokines. Those cytokines are then going to interact with the B cell, and it's going to initiate or activate that B cell. So now we have an activated B cell, and then that B cell is now activated, and it's going to make many copies and differentiate. Okay, so up here, the B cell interacts with the pathogen or free antigen. It's going to internalize it and then display the antigen with the MHC class 2 molecule. The B cell is then going to bind with that T helper cell that also matches the antigen. Then the T helper cell releases the cytokines that are then going to activate that B cell. So then if we look further down here, these are the images we've already taken a look at. So now we've had that cytokine, or the cytokines are released. They're going to go over and activate the B cell. This activated B cell then is going to divide to produce plasma cells and memory B cells. You can see now it's activated. We go down here. Now we are going to make plasma cells and we are going to make memory B cells. Remember that memory cells are so that they are floating around in the bloodstream. We have lots of copies just in case we encounter that pathogen again. Once we do, then we have lots and lots of memory cells around so that they can take care of it very quickly. Now our army of plasma cells are those that are going to be secreting large amounts of antibodies. And again, those antibodies bind to the pathogen and do the various things we've spoken about previously. So our B cell that's been activated by the T cell, by the cytokines of the T cell, are going to produce memory cells to help us remember later. And they're going to make a bunch of plasma cells. Those plasma cells then are making these antibodies. Remember that antibodies neutralize the pathogen, they activate the complement cascade, and they cause agglutination, so a clumping up of the pathogen. And remember, part of this neutralization we have is opsonization as well. So it makes it easier for phagocytic cells to grab on to that pathogen. So our plasma cells are just, little machines, little factories for these antibodies. Once we have this cell that's been activated, it's activated given this antigen piece. So when it makes copies of itself, it's making all of these plasma cell copies that have that exact same receptor and have interacted to that, which means that it's going to be producing antibodies to fight against that particular pathogen. So then we have our T cell independent activation of B cells. So what we just looked at was T cell dependent. We have to depend, that B cell has to depend on a T cell in order to become activated. In this case, we're going to see T cell independent activation. So it doesn't have to have a T cell in order to be activated. In this case, we have the B cell receptors going to bind to the non-protein epitopes on the pathogen, because remember, when we're talking about protein epitopes, we're talking about T cell activation, T cell dependent. If we're talking about non-protein, so lipids, carbohydrates, nucleic acids, then that B cell receptor is going to bind to those. That's going to be the first signal. Now the second signal is required, and that comes from interactions with toll-like receptors or PAMPs, or other factors from the complement system or the complement cascade. What these do then is they activate the B cell. Once the B cell is activated, then it divides to produce those plasma cells. So it's the same as the second portion of what we just spoke about. So here the polysaccharide antigen with the repeating epitopes on it. Then we have our pathogenic bacterial cell, of course, but then we have our B cell over here. You can see that it is binding to this polysaccharide antigen here in a couple of places with its receptor, remember, which is an antibody. So it's going to attach to that polysaccharide antigen that has these repeating epitopes, and then that is our first signal. So down here what we're seeing is the first signal, which means that we have to have two things that are going to cause the B cell to become activated. When we just saw the T cell dependent activation, our B cell interacted with the pathogen. That's the first one. The second one was when it interacted with the T cell, and the T cell produced cytokines to then fully activate it. That was the second response, was the interaction with the cytokines from the T cell. In T cell independent activation, what we're seeing here in this illustration is the first signal. The B cell is going to interact with the polysaccharide antigen or nucleic acid or whatever, and then it has to wait for a second signal. So once that second signal happens, then we have our army of plasma cells is going to be produced. And you can see that over here on the right side. So our T cell independent B cell activation, once it's activated, we have our B cells that are going to produce all of these different antibodies. So the example they're showing here are the IgM antibodies. Remember, IgM is something we can use as a diagnostic tool because as soon as we are infected with something, we see a big spike in IgM. So our B cell is going to interact with our polysaccharide antigen, and then it's going to get its second signal from something else, right? So not a T cell. And then it's going to differentiate. So we're going to, or it's going to clone itself, make many copies. So it's going to make lots of plasma cells. Those plasma cells are now going to be secreting antibodies that'll bind to the pathogen and kill it, hopefully. But remember, by killing it, I mean neutralize it, activate the complement cascade, cause agglutination, opsonization, those things. In this case, it does not result in the production of memory B cells. This is a very short-lived response. So when we're talking about T cell independent activation, this is kind of the lesser of the two because we don't get that memory like we do with the T cell dependent activation. In T cell independent, we're not communicating with the T cell piece of the immune system. So then it ends up being just the B cells that are trying to fight off this particular pathogen. All right, so let's put this together with our primary response. So our immune system response after the first exposure to a protein antigen. Right, so we have a primary response is when our body is going to be exposed to a protein antigen. And then we have our T cell dependent primary antibody response. So here, again, we have our antigen on our bacterium. Our B cell here is going to pick up on that via the B cell receptor. Then it's going to present a piece of the antigen on its MHC class II molecule. Then we have our helper T cell come along, recognizes that, releases those cytokines that are then going to activate that cell. So then when that cell is activated, remember since this is our T cell dependent activation, then it's going to differentiate into memory cells and plasma cells. So then the plasma cells are going to release lots and lots of antibodies to help to try to get rid of this particular pathogen. And the memory B cells are going to hang out for next time it sees it. So the initial stage is a lag period, right? So our immune system is going to be exposed to a protein antigen. Then the T cell dependent primary antibody response happens. So this whole thing here, we activate our B cell divides, and then we start to make plasma membrane or plasma cells, but they don't immediately start making antibodies. They have to start going through the process, right? So they have to get the resources and start to build those proteins before they can start to release them. Now, this is our lag period. This is where no antibody can be detected in the serum. So it takes about 10 days after being exposed to a particular pathogen for our bodies to start producing the antibodies to fight it off, and then eventually go around and start fighting this off. So all the steps of the primary response are occurring at this time. So a lot of those things that we already spoke about with inflammation and fever and things like that, they're going to just be starting. So the end of the lag period is marked by a rise in IgM levels in the serum. So remember, IgM was good for diagnostic for illness. So we have our primary antigen exposure here, and then we have time going in this way, and then we have the antibody concentration in the serum. So we have this lag phase, and it's a lag phase until we see this rise in IgM. So our lag phase goes to about here, let's say, and then we have the rise in the IgM. Once we have that rise in the IgM, then we say, okay, this person actually is fighting off an infection or some sort of pathogen. Then as IgM levels decline, so after we have our first exposure, so we have a high amount of IgM, but then they're going to decline, and we have our IgG, which are always in the bloodstream, that is going to increase at that point. So IgG levels rise until peak is at about 3 weeks. So it takes quite some time for us to be producing those IgG cells. And then we have our memory B cells that are produced for our secondary response. So you can see that we have some overlapping with the antibodies here, which is good because we want antibodies, or we want our antibodies to be available for the particular pathogens. So here we can see in this table why memory B cells are so effective, or how effective they are. So we kind of took that illustration or that graph from previously. This is our primary antigen exposure. So when we're exposed to something that we've never ever seen before. Then we have that lag time where we start to see the IgM, or up until we start to see that IgM increase. Remember, this is the IgM antibody. So that increases, which is going to increase the chance that it bumps into some of these pathogens and they start to destroy them. But then they are going to go down. When the IgM antibodies go down, again, our IgG, which were pretty low, are going to go up and peak. And then eventually come down, but at that point, then you are starting to feel better. Now, remember that when this happens, we have a faster and stronger immune system response when we're exposed to something the second or further times. So here they're showing that this is where this person was exposed to that particular antigen again. So then we can see here, we still have our relatively steady IgM antibodies here, where we've got a lag time here before we have a whole bunch of them in the bloodstream. However, if you take a look at the time frame here, our lag time over here, even if we went to the very beginning of our IgM peak, if we went to the very beginning of our IgM peak over here, each of these lines, that's 5 days. So this would be 15. So let's say this is about 12 days. Over here in our secondary response, we respond to it, and then at first we have that lag period, and then we have the beginning of this IgM peak. This space, you can see here, this is maybe 12 days, and over here we're looking at maybe 4 days, because this is 10, this would be 5, and then down here would be 0. And then you can see we have our regular IgM peak and then goes down, but our IgG gets really, really high. And that's because we have so many of these memory cells that are floating around still that they immediately are going to start making antibodies for it. And that's why we get so many IgG and why the time frame is so much shorter. So the lag period can be a few days, and then we have a production of the IgG is higher after that. Antibodies are more effective and bind with higher affinity. And this is why when we have our memory cells and they're producing all of these antibodies, that they are most effective and they bind with higher affinity. So they can attach, and they don't have this giant cell attached to it or anything. So it can have these antibodies attached. And so they're more effective this way. So we can see why we have this huge IgG peak compared to our primary antigen exposure. And then plasma cells that are produced are longer lived. So when we do produce our plasma cells that are pumping out all those antibodies, they live for much longer. And that's why we can have all of these antibodies and we can have hardly a dip in antibodies. And that's because we have so many of these memory cells that are floating around in the body that we get this really large immune system response right away. Which is helpful because then we can fight things off much easier and we may not even feel sick.